We are Science for Life. We are here to make the world a better place. We are the trusted ally of animal producers in Asia, Africa, and the Americas. We are here to create value with sustainable, efficient, and innovative products that transform challenges into effective solutions. Solutions for animal welfare, solutions for you. Smart and advanced products to take care of the productive development of animals. Natural alternatives for conscientious production and development. Functional additives to enhance productive performance. We are proud members of Illuma Alliance, a place where knowledge, technology, and innovation come together to forge a better world. Our innovation centers help us validate the safety, feasibility, utility, and sustainability of new concepts. Our international laboratory network allows us to provide ultra-precise and efficient solutions. Our pilot plants enable us to transform concepts into functional products. Our experimental farms offer precision in vivo utility testing. This is our ecosystem of skills, capabilities, and mindset. This is the ecosystem we make available to you in order to provide solutions for your animals and for a better world. We are Allura. Science for Life. We are Science for Life. We are here to make
Good morning, everyone. Welcome to the Allura Looking Ahead webinar, Protecting Your Flock, Addressing the Enterococcus Challenge in Poultry. My name is Cristina Mesa, and I am part of Allura's marketing team. Today, I have the pleasure to be your moderator for this webinar. Before we start, we would like to inform you that this webinar is being recorded and that we will have time for questions and answers at the end of the presentation. Feel free to post your question in the question box, and we'll do our absolute best to get to as many questions as possible. Today, we have the honor of presenting Dr. Matthew Kirkland-Jones and Mitsu Suyemoto. Mitsu Suyemoto has been a researcher in North Carolina at the North Carolina State College in veterinary medicine for over 35 years, working with multiple groups through the years. The overreaching emphasis of her work has been on bacterial and infectious disease, molecular microbiology, and animal infection models with a primary focus on Enterococcus equorum, Enterococcus faecalis, Enterococcus coli, and Salmonella in poultry. She is proud to have supported the poultry industry in her research efforts, and as an avid mentor of numerous residents and graduate veterinary students, she has contributed to the academic of professional development of the next generation of veterinary excellence at North Carolina State University. Mitsu, thank you for being here. Dr. Matthew Kirkland-Jones is a poultry scientist based in Athens, Georgia, specializing in veterinary medicine and nutrition to conduct applied poultry experiments. Originally from Thomasville, Georgia, he studied avian biology and earned a DVM and PhD in poultry science from the University of Georgia. His research focuses on feed additives and poultry health. Matthew has served as Director of Research and Azomite Mineral Production and now works as SPRG. Focusing on applied poultry research outside of work, he enjoys spending time with his family, gardening, and being outdoors. Dr. Matthew, thank you so much for being here. Now I'll pass the microphone to Mitsu. Mitsu, the stage is yours.
Thank you. Let me share my screen with you. All right. Right. Good morning. Today, I'm thank Allura for allowing us to present this webinar. I'm going to talk to you today about pathogenic Enterococcus secorum and share with you some of the things we've learned over the past almost 15 years now, and some of the things we still don't know. And I hope this will provide a good review and an introduction to the studies that Dr. Matthew Kirkland Jones will tell you about. So before I even get started, I want to recognize Luke Borst. And Dr. Borst's observations and curiosity are what launched us into this Enterococcus sequorum research mode. And Dr. Grayson Walker, who completed his DVM and PhD in our lab, was also part of this work. And his thesis focused on Enterococcus faecalis and its interactions with avian pathogenic E. coli. So today there's a brief introduction. Talk to you about the difference between commensal and pathogenic Enterococcus sequorum, show you our disease model, the experimental models we use in our work, and then use those models to talk to you about the different clinical presentations that you're finding in the field now. Then I'd like to switch gears and talk about possible interventions. And if there's enough time, I'll briefly touch on Enterococcus faecalis and poultry disease and share with you some of Grayson's thesis work. So to start, Enterococcus sequorum was first described as a strep in 1983. It's a major component of the gut microbiota in adult birds. They are gram-positive cocci that grow very nicely on blood agar. They're lactic acid bacteria, which means they not only produce lactic acid, but are very happy growing in low pH conditions that are along with lactobacillus, etc. And that's really all we knew about Enterococcus sequorum or even thought about it until 2002, when there were multiple reports of birds in these strange postures. These birds were paralyzed with their legs cranially extended. And at necropsy, they found this very distinct lesion in the free thoracic vertebra of these birds. And that lesion cultured pure Enterococcus sequorum. So this disease, enterococcal spondylitis, was first described in Europe in 2002. And we didn't have cases of it in the US until 2009. And they were first found in North Carolina. They're often still called by the misnomer kinky back, which is actually a congenital deformity of the spine. It affects broilers and broiler breeders. We see a sepsis and a pericarditis at 2 weeks of age, and that's followed by paresis and paralysis starting about 4 weeks of age. And you can see in that lower frame how the lesion impinges upon the spinal cord. The mortality is variable, roughly 2 to 15%, and to date, there's no effective antibiotic-free treatment. So what had changed? Had something changed with the birds, or had something changed with the bacteria? So to answer this, we started off by doing longitudinal field studies. At affected farms, we collected gut, spleen, and spine starting in week 1 and continued for 6 weeks. And as you can see in this graph, already in week 1, we were able to isolate Enterococcus sequorum, not only from the gut, but also from the spleen. And by week 3, we were starting to find positive spinal cultures. But on the control farms, on the farms that did not have an outbreak, we weren't able to culture Enterococcus sequorum at all until week 3, and even then, only very small amounts and only from the gut. And we were never able to isolate it from spleens or spines. So what's different about those bacteria that we were able to isolate in the first 2 weeks during an outbreak? Well, we started by doing pulse field gels, and these are just DNA fingerprints, so you look at the similarity of the banding pattern. And you can see with the pathogenic strains, they were very similar to one another, while the commensals had a very different pattern. And these are almost clonal with a 90% similarity. And we took this one step farther and did whole genome sequencing. We found that there were genes that these genes that are in red that were conserved in the pathogenic strains, but absent or very different in the commensal strains. And most of these genes have to do with capsule or cell wall biosynthesis. And just to make sure that this was not something unique to North Carolina, we then followed 22 distinct outbreaks in the US. And again, we cultured spines and spleens, and we found that this same pattern held up. They were very similar to one another. And again, the whole genome sequencing, we found that they were the same patterns held, the same virulence genes appeared. And in fact, we focused in on the CPSC region. And those are the capsule synthesis genes. So again, these genes that were unique in the pathogenic strains mostly were in the enterococcal cell wall or this outer capsule. And here you see the lipoproteins, lipoteichoic acid, EPA polymers, and this capsule. So we decided we would knock out the CPS locus, part of this capsule synthesis pathway, and the CPS gene, which is in this variable region downstream in this capsule synthesis pathway. And when we knocked them out, we found that the deletion mutants were indeed attenuated in both our embryo lethality assay and our bird challenge model. And I'll talk to you more about these models in a minute, but suffice it to say that we had no sepsis with either of these knockouts. While we had, with the parent strain, we had sepsis and spinal lesions. So we know that these two genes are at least in part responsible for the virulence of the pathogenic strains. So let's talk about our disease model. If we go back to that time course of pathogenic EC infection, we see again the gut, the spleen already colonized in week 1. And then we eventually develop these spinal lesions. So here it is in a different form. Our disease models that the gut is colonized initially in weeks 1 and 3. The birds become septic, and eventually enterococcal spondylitis sets in, and the paralysis is seen. So, we often have the question of what is the role of gut injury? And we have found that the gut barrier damage is not required for the Enterococcus to escape. And coccidiosis does not increase the EC sepsis. And in fact, in an experimental challenge we did, the EC sepsis and the spinal lesions actually decreased in the presence of a severe Eimeria infection. And I cannot explain those results. Another question we frequently get is why this free thoracic vertebra? Why is the site of the lesions? And you can see in this picture that this particular vertebra undergoes a lot of mechanical force, especially in these big broilers. And what we learned was that osteochondrosis dissecans, or OCD, predisposed the birds to enterococcal spondylitis. So in this first pane, you can see it with the arrows, there are cracks in the cartilage. And once the birds are septic, these cracks become colonized by bacteria. And as the bacteria multiply, you get disruption of the cartilage and the bone. And you can see this is already impinging on the spinal cord. And the same process holds true for the femoral head necrosis that we see in these birds. So let's talk about our experimental models now. The first model that we frequently use is the embryo lethality assay. And this is part of the 3 R's use of in animal research. So we have replaced birds, we've reduced the number of birds we use, and we've refined some of our research protocols. And we use this assay to compare the virulence of field strains, to look at the some of our genetic mutants, and also to look at co-infections of different strains. So we incubate eggs for 12 days, and on day 12, we do an allantoic injection with 10 to the 2 CFUs of our strains. We then candle the eggs every 24 hours for 5 days, and we record mortality, and we culture all the mortality and the surviving eggs to make sure everybody got what they were supposed to and there are no contaminants. And when we do that, we end up with a Kaplan-Meier survival curve. And this is the very first one we did. And we found that isolates from the gut had about a 65% survival rate, whereas the embryos inoculated with isolates from the spine had only a 25% survival rate. And you can see the embryos from the eggs inoculated with the commensal strains look very similar to the eggs that were the embryos from the eggs inoculated only with PBS, whereas the embryos from eggs inoculated with the spinal strains are very hemorrhagic and edematous. Our second model is the broiler challenge study. And depending on our power calculations, we usually start with 120-day-old broiler chicks per group. They are assigned to PVC floor pens with shavings, and there are 20 birds per pen. On day 1, usually, we gavage them with 10 to the 7th CFU of the challenge strain, or PBS. And this time can vary up to day 3, 5, 7, depending on what we're testing. We then sample spleens from 4 birds per pen on day 14 post-inoculation. And then at 35 days post-inoculation, we culture spleens and spines from the remaining birds. We also culture all mortality. So I'm going to use those 2 models now to talk about the strains we're finding in these pericarditis and early mortality birds. So if we go back to our original weekly mortality pattern, this is from 2017, and we saw a small peak of mortality in affected farms from weeks 2 to 4. But then once those birds had spinal lesions, we had a much greater peak between 4 and 7 weeks. And these birds, of course, were called, or they died from starvation or dehydration. And this is the paralysis presentation. But more recently, we've seen a different pattern where we see an increase in mortality from weeks 2 to 4. There's still mortality later on, but we've lost many of these birds already at this point. And this is the sepsis presentation. So again, we ask, what's changed? Is it something with the bacteria, or is it something with the birds or the management? So again, more pulse fields, more DNA fingerprints. And these were from samples that were collected in 2022. So we looked at an 80% similarity cutoff. We see that the hard strains all grouped together in clade 3, whereas if you look at clade 1, that's where most of the spinal isolates reside. So we know that they are genetically distinct from one another. And again, our next step was to do whole genome sequencing. And in this image, you're looking at the percent of protein homology. And the more purple you are, the more similar you are. Inside here is our standard spinal isolate. The next 2 rings are isolates from the heart. And the outer ring is a commensal strain. And when we first did this, we were a little confused. The heart isolates look very similar, more similar, in fact, it seemed, to the commensal isolate. I was like, what's happened? But when we looked closer and did genomic analysis, we found that indeed the heart isolates had acquired many of these same virulence genes that these original spinal isolates had. So it seems those genes had moved into a commensal strain. And some of those shared genes were, again, those capsule biosynthesis genes. And when we used the heart isolates in an embryo lethality assay, we found that their virulence was slightly less than the original spinal isolates. Here we have 5759, and the survival rate of that is the same as the commensal strain. About 75% of the embryos survive. The 5872 heart strain was slightly more virulent with about 60% survival rate, whereas in this study, the spinal isolate inoculated embryos had only a 50% survival rate. We then moved on to doing a bird challenge, and the same prevalence of spleen infections were seen in the birds. So Again, the spinal isolates had the greatest prevalence of sepsis. Then the 5872 was lower, and 5759 was yet even lower. And that same pattern again showed up in the prevalence of spinal lesions. And strangely enough, it also, that same pattern of prevalence was found in the with pericarditis. And we expected the opposite of this. We expected higher numbers of pericarditis with the 5759 and 5872, but that's not what we found. So from that study, we concluded that the heart strains are genetically distinct from the original spinal isolate pathogenic EC. We know that the heart strains harbor virulence genes, including those required for capsule production. Those heart strains are pathogenic in both our embryo lethality assay and our bird challenge model, although not quite as pathogenic as the original spinal isolates. And the factors that predispose those broilers to early disease require further investigation. I have some feelings about the bug side, but no facts to back them up yet. So let's move on and talk about possible interventions. When we talk about interventions, everyone always immediately thinks, oh, a vaccine. A vaccine would be great. We could produce antibodies, and so you could neutralize the bacteria as soon as they escape from the gut before we set up these septic spleens. It would be wonderful. We could prevent the spondylitis and the paralysis. But there are problems with this vaccine approach. First of all, immunity has to be present in that very first week, so that you could never vaccinate the broilers themselves, possibly the breeders. But antibodies that are produced in response to the vaccine strains may not be protective. And we know the extensive capsule of pathogenic Enterococcus may evade phagocytosis. And in fact, we know that to be true because we've done opsonophagocytosis assays with elicited macrophages, coelomic macrophages from the birds. And when we expose those macrophages to commensal strains, whether or not they've been opsonized or not, they're consumed, they're phagocytosed by the macrophages. But if we use pathogenic Enterococcus cecorum, these are two different strains of the pathogen. Even if they're opsonized, the macrophages do not see them. So that capsule is offering up that perfect cloak of invisibility. So they escape phagocytosis completely. So a rationale for the monoglycerides or DFMs or probiotics is, can we actually inhibit the bacteria before they ever leave? Can we prevent colonization of the gut? And the hope is that the monoglycerides will help neutralize these bacteria so they cannot colonize the gut. And the use of DFMs or probiotics is that they may outcompete these pathogenic bacteria in the colonization. And if either of these two are effective, we could then reduce colonization, reduce the sepsis, and reduce the spondylitis. And if anybody has worked with these birds, they know that the welfare of these birds is important, and we would love to be able to reduce the spondylitis. But what if we could take an even earlier approach? We now know that we can find pathogenic Enterococcus cecorum in the hatchery, in the transfer residue. So these are our first knowledge that this bacteria is vertically transmitted. But we don't know if these bacteria are on the surface of the egg or actually in the egg. So if we go back to those pulse field gels and we look here in clade 2, we've had identical isolates from hatchery residue, and then they were same identical bacteria were collected from birds downstream. Birds that have been set at 2 weeks, we found heart and air sac lesions in birds. So we know it's in the hatchery. Could we monitor the hatchery? Could we monitor the transfer residue and then work back to the breeders, the grandparents, and the pedigree birds? Would this be a way to eliminate Enterococcus faecorum? So in summary, the pathogenic Enterococcus faecorum strains have acquired specific virulence genes, and we can use those genes to distinguish pathogenic strains from commensals. The strains we've tested the pathogenic strains can cause both pericarditis and spinal lesions. The bird or environmental differences may contribute to the increase in this pericarditis presentation. That remains unclear. And hopefully the interventions may decrease the prevalence of the disease and improve the animal welfare. And then we come to the question, is eradication possible? With a lot of work, I think it could be. So I'd like to acknowledge our industry partners for generously supplying us with eggs and chicks throughout the years, and then U.S. Poultry, USDA, the NC State College of Veterinary Medicine, and the Southern Poultry Research Group for supporting our work. These are our references, and if I can also now, if there's enough time, go ahead and speak a bit about Enterococcus faecalis. We'll just touch on it very briefly. We know that Enterococcus faecalis is a much bigger problem now than it used to be. We know it's being found in the hatcheries, and our hatchability is now down to 80%, roughly. We know Enterococcus faecalis is not the only problem giving us that lower hatch rate, but we know it's a part of it. We know there's also increased early mortality in the chicks that come from those hatcheries. And when we culture them, we find positive yolk sac infections. They're positive for Enterococcus faecalis. And often they're found with E. coli, with APEC. We also know that Enterococcus faecalis is responsible for amyloid arthropathy. So a lot of work is now being done on Enterococcus faecalis, but I'm going to share with you some of Grayson Walker's thesis work on the co-isolation of APEC and Enterococcus species. And almost all of those Enterococcus species are Enterococcus faecalis. So Grayson looked at 6 different flocks. They were our teaching flocks, 3 turkey and 3 broiler flocks. And we looked at mortality due to colibacillosis. And when we looked at co-isolation of Enterococcus faecalis with the avian pathogenic E. coli, we found that in 8 to 100% of the cases, we had both. And so Grayson's hypothesis was that Enterococcus faecalis enhances APEC virulence. So again, our first look was in our embryo lethality assay. You see here that Enterococcus faecalis had a 75% survival rate. The APEC alone had about a 50% survival rate, but when we combine the two in a co-infection, there was less than a 10% survival rate. So again, it seems that the combination is worse than either of the single isolates. So now I want to set this experiment up for you. So we know that avian pathogenic E. coli needs iron to grow, and many of its virulence factors are indeed iron scavenging proteins. So in the yolk sac, as the egg matures, it's a very iron limited environment. So Grayson, this is simulating a Petri dish. And so he made media that was also iron restricted. And he then dotted the avian pathogenic E. coli, these are the pink spots, in increasing proximity to the Enterococcus faecalis. And the theory being that if the Enterococcus faecalis is secreting something that can move through the agar, that you will have a greater effect where the colonies are more proximal. So Grayson set up a grow camera in the incubator and followed these colonies for 5 days. And I'd just like you to watch this for just a minute and keep an eye on the apex colonies closer to the Enterococcus faecalis. Whoops. So at 48 hours, you can already see a difference. And at 120 hours, you can see a big difference. And we know that that's not only an increase in growth, but an increase in the expression of virulence factors in the avian pathogenic E. coli. So we know when you find the two together, it's not good. So one more thing I'd like to touch on is amyloid arthropathy. We think of that as a disease of layers, not broilers, and we think of it as a disease that's only found in Europe. There's been one case in California many years ago. And in this picture, you can see the orange amyloid deposited in the joints of this bird, and which is pathognomonic for this disease. And our current poultry resident, Dr. Aaliyah Gore, just recently completed a study with a pullet breeder layer flock in Ontario, Canada. This flock they were poor doers from the very beginning. They had increased mortality, growth depression throughout the life of the flock, and sporadic lameness. And when they cultured these joints, they were able to isolate Enterococcus faecalis. And we did whole genome sequencing and were able to find sequence types ST82 and ST49, which are known to cause amyloid arthropathy. So this has now appeared in Canada. So it's something else the industry might want to keep an eye out for. So that concludes my talk, and I would like to thank Laura and turn my screen over to Dr. Matthew Jones.
Thank you so much, Mitsu. That was a great presentation. Dr. Kirkland, the stage is yours.
Excellent. Thank you so much. See if I can shift over here. Is that coming through for everybody? And I guess yes, unless I hear otherwise. Yeah, so thank you so much for that introduction, Mitsu. That was fantastic. I always learn something when I listen to your presentations, and thank you, Allura, for inviting me to be here today. So just to recap the introduction, my name is Matthew Jones. I'm a veterinarian with a graduate degree in nutrition, and I work at Southern Poultry Research Group, where we do applied health-based poultry research. And if we're to kind of categorize the studies we do, it's predominantly bacterial and protozoal, specifically Amelia challenge models that we conduct. And about half of those are enteric health in nature. And then the next largest category is food safety. But if I was to pick a bacterial disease that's increased relative to the others as far as interest in modeling, I'd say certainly over the last couple of years, it's been Enterococcus. So, 4 years ago, we were doing a couple of studies a year, and then it seems like it's really exponentially increased over the last 3 or 4 years. So I'm excited to be here today, giving you some of the insights from my end on how to manage this disease in terms of biosecurity and some of the products that we've evaluated here recently. So just to recap again, my background so you can understand the perspective a little bit more. So I came to the University of Georgia for undergrad and then transitioned straight into really a dual DVM PhD program where I did vet school during the academic year, and then over the summers I did graduate work until I finished vet school and then focused on my PhD in applied nutrition and intestinal health. I did spend a year at Azomite Mineral Products where I really met the industry and got to learn a lot of things, including feed manufacture. before I transitioned into my role here at SPRG, which is predominantly as the director of nutrition. And I'm often the attending veterinarian on the experiments that we run. So in the morning, I tend to focus on veterinary tasks like gavage, lesion score, necropsy, and then some of the technical sampling that we'll talk about in a minute, like the spleen collection, and some of the samples that we will evaluate moving forward. So as far as the breakdown of today, we're going to speak a little bit about applied observations, or really as applied as I can be in a controlled experimental context, but really from what I'm seeing, what implications that might have on those facing the disease in the field. Similarly, some comments on biosecurity, given what we observe in our research setting that might have impacts on those trying to manage the disease. And then lastly, the last half of the presentation, we'll talk about some of the interventions that we've evaluated here at SPRG. So as far as applied observations, these really are the topics that I want to discuss as we move forward. So it's going to be percent mortality, and this is strictly SPRG modeling, and this is the kind of the lens that this data is going to come to you through. So percent mortality, the percent of birds that present with clinical spondylitis in the challenge models that we run, the percent of suspect mortality that's cultured. So oftentimes we'll collect samples from those that have clinical lesions, specifically pericarditis, parahepatitis, femoral head necrosis, or free thoracic vertebra infection. And so those are going to be cultured. And so we'll break that down. And then the percent of the cultured mortality that comes positive for Enterococcus when we submit it to the laboratory. And then also we'll pair that with the percent positive prevalence for free thoracic vertebra. And I'll try to remember to mention the spleens as well, that we often see at 42 days. So exactly what Mitsu was describing in the progression of the disease, we tend to see more septicemia early. So 2 to 3 weeks, we'll see more pericarditis, parahepatitis, potentially ascites as a complication of that. And then later in the infection process, really around 4 weeks, we'll start to see leg issues. And the reason that I chose the femoral head for my example of leg issues is because that's what we often see in our challenge scenario. So really, as far as percent of clinical enterococcal spondylitis, I'd say it's probably around 1%. Most of these birds are not presenting with that classical kickstand positioning. We do see a lot of lameness, but it usually comes back to either femoral head necrosis, unilateral or bilateral femoral head necrosis in those birds. And again, you can see this lame bird in this image. We don't see as much of the classical lesions, but what we do see are really devastating impacts on performance. Mitsu mentioned this as far as welfare is concerned. You can visibly see the uniformity issues in the birds displayed in this picture. And so we do see negative impacts on body weight gain and feed conversion in our modeling. So what I did for the sake of kind of showing everyone what it looks like in experimental conditions is I picked 5 different studies that we've conducted over the last several years. And I didn't have any particular criteria to pick these studies other than they are not deviating from the normal. So if we did something a little bit different with the modeling, those would not have been included. And so in these studies, you can see our free thoracic vertebra prevalence is between 10 and 20% positive prevalence at 42 days. And if I was to include spleens in this, in our challenges, it's probably between 40 and 60% on average positive prevalence at 42 days in the spleens. But as far as mortality, I'd say it averages between 4 and 6%. Because of the types of modeling we do and the handling routinely, it's not uncommon for us to see 5 to 6% mortality in the challenge models that we run, and even the non-challenge models that we run. And so you can see kind of the average there is just over 6%. So this would be kind of a high normal mortality in our challenge context. But we do see a good bit of we do see a lot of clinical signs. So the next column there is the number of mortality that's positive for Enterococcus cecorum. And again, these would have been birds exhibiting signs like pericarditis, parahepatitis, ascites, femoral head necrosis, free thoracic vertebra abscess. But that would have only represented maybe half to a third of the birds that we are evaluating. And you can see based off these numbers, 16 of the 75 in this situation were positive or just at 20%, right? So if we're sampling half the birds and 20% is positive for Enterococcus, right? We have a 1 in 10 chance based on the mortality of detecting that pathogenic Enterococcus. And so what I'm trying to get at in this description is the conditions can be a little bit difficult to interpret. We have just above normal mortality in our challenge context, and then we have a little bit of lameness increases, but when we culture those birds, 10% are coming back positive for Enterococcus. So it's a little bit obscure, really. I kind of wanted to digress just a second and tell a story. So on myself, I guess. So I do a lot of the technical sampling, and I go through and do the mortality either at the beginning or the end of the day. And when we're doing Enterococcus studies, sometimes we'll have a bird that presents classically with like a bilateral, excuse me, a bilateral femoral head necrosis. And so I'll open the bird and I'll take a transport swab and I'll swab the femoral head and I'll take it to our lab manager and fairly confidently say, this is going to be Enterococcus, and I'll kind of flop it down. And the next day, equally as confident, our lab manager will say, no, that was definitely E. coli. And so that kind of from a clinical standpoint, I could see that further obscuring the outcomes. And based on what Mitsu was saying, I don't know if that's the relationship between the E. coli and the Enterococcus, and that's just happenstance, but it is kind of interesting. Just that it is usually E. coli that comes out of those when I am so confident there. So yeah, so it can be fairly obscure disease, and it could be frustrating, I could imagine, in the field to diagnose this, especially if you're not using samples from the spleens and the free thoracic vertebra of birds around 42 days in live birds. So I want to make a comment on biosecurity because I think it's could be important for those dealing with this disease in the field. So most of the models, like I mentioned, that we conduct are bacterial challenge models. And so we are very cognizant of biosecurity in all of our research spaces. So we boot in, we step into dry chlorine bath or powder baths. And then when we go into the birds, we usually have PPE assigned to that room. We're wearing PPE, and then we're going to step into boots that are assigned to that particular pen. And so we take a lot of precautions as it relates to spreading the disease or biosecurity. But the example that I'd like to tell you about was actually in an isolation room. So isolation rooms, we take the biosecurity up one more level. So this has a single animal caretaker. Otherwise, there's a camera in the room that those passing through, like me, can observe the birds on. And they have to boot into an anteroom, put on PPE, and then put new boots on as they step into the birds. And so that's kind of how this space is structured. And within the room where the birds are, the walls are solid sides. So not only are the external walls solid, the walls between the birds are 24 inches high, and they are solid sided. And so what we did was a model using SA3 and the 5872 pericardial strain that Mitsu mentioned in her portion of the presentation, relative to an unchallenged control group. And so treatment 1 there is an unchallenged control. Treatment 2 is the enterococcal spondylitis isolate, SA3, and then the pericardial strain as 5872. And just to describe that a little further, they would have to step into the 5872 pen and then boot into the SA3 pen, but to work those birds, they didn't actually have to step into the unchallenged control. So despite that, at 10 days, you can see this prevalence data, the unchallenged controls were already positive for Enterococcus, similar to the SA3 challenge, actually. And if we follow that to 14 and 17 days, it tracks fairly well with the SA3 data. Again, this is only 5 birds at each sample point out to 21 days. And then at 21 days, we collected all the remaining birds. And so that unchallenged control remained consistently high, despite not receiving a direct challenge. And so this is the example that has been published, but this is something that we've observed in most of the modeling, really all of the modeling, where the birds are confined to the same airspace. If there's an unchallenged group, eventually that Enterococcus will spread. And so that, I think the implications there from the field side would be on sampling and intervention strategies, because I think if you think you have Enterococcus, it is likely that the whole house has experienced or will be experiencing that pathogenic Enterococcus. Again, I think that would be important for those sampling and or trying to intervene. Yeah, and so speaking of intervention, that's probably a fair transition into interventions. So we're going to walk through some of the strategies that we've evaluated and published on in the last couple years, but first I'm going to revisit the modeling that Mitsu already introduced. And really at SPRG we focus on that model where the birds go out to processing age. And so what it looks like is we receive male Ross broiler chickens, and then we spray vaccinate them with a commercial coccidiosis vaccine. So the way that we conduct our model is we receive male Ross broiler chickens from the hatchery, and we spray vaccinate them with a commercial coccidia vaccine. We allow those birds to preen and receive the vaccine and dry. Then we'll weigh them and allocate them to the pen. Now, just like Mitsu was describing, usually we'll challenge them from 0 to 7 days. Often it's 0 days. And so just to kind of explain that process, after they're weighed, they're allocated to a pen. And then one of the technical people on our team will come through and gavage 0.1 mils of Enterococcus aucorum at a dose of around 10 to the 7 CFU per bird to each bird, and then place them in the pen with the fresh pine shavings. We usually use 6 to 8 replicate pens of 25 broilers. And as far as the sampling, that kind of changes how we do things. And we'll kind of transition into the sampling from here. So we often take that early septicemia evaluation that Mitsu described from 14 to 21 days, just depending on the goals for that given study. And then at termination, which could be anywhere between 35 and 42 days, often at 42 days, we'll collect spleens and free thoracic vertebra swabs from 100 representative birds. And so it depends on the model as far as the number of replicates we have. Say it's 8. If we take 100 birds, we'll end up taking 12 out of 4 of the pens and then 13 out of the remaining pens, and just to hit that 100 representative birds. Then once we've collected those samples, we send them to Mitsu at NCSU, and she tells us if they're positive for Enterococcus. And then we send that information off to Dr. Roy Burgess, who is a biostatistician at the University of Georgia, and he differentiates whether there are differences between the treatments. So the first intervention that I'd like to describe is vaccination, and Mitsu's already mentioned it. But just like she was describing, it would be fantastic if we did have a vaccine as an option, because it makes a lot of sense. There are other bacterial diseases that can be mitigated to some extent with vaccination fairly effectively. But up until this point, as Mitsu mentioned, there has not been effective interventions for vaccines. And so this is a study that the Bourse Lab published at NCSU, looking at different challenges after they've been vaccinated. This would have been the breeders have been vaccinated, and we're looking at the offspring prevalence. I can't in this moment remember what the vaccine was homologous to, but there was a homologous challenge and a heterologous challenge. So one of the isolates was used and challenged directly versus one isolate was different from what the vaccine was actually made from. And as you can see in the outcomes, we have the placebo or the sham there. So that would have been the unvaccinated group relative to the vaccinated group, and there were no significant differences. But again, it would be phenomenal to have that as an option to produce for producers out there. The interventions that we've evaluated tend to be added either in the feed or at hatch. And so the first intervention that I'd like to describe would be monoglycerides. And so in the last year or so, we have evaluated monoglycerides, and because of the efficacy of these products, we have presented both at PSA and AAAP this past year. So we're going to focus on the PSA presentation. This is Allura's product. This is a blend of monoglycerides called BIMU. And so in this series of experiments, we started in vitro and kind of worked towards a more practical situation. So we're looking at optical density there on the y-axis, and then time on the x-axis. And the way that we do these evaluations is on a plate reader, and we're looking at a specific wavelength of light. And so the more dense the growth is, the less light penetrates. So you can see that darker navy line there showing more dense suspension versus all of those BIMU concentrations there at the bottom, having a substantial impact on the growth of that culture. And so it's fantastic to have in vitro data. It gives us a lot of information and helps us make informed decisions. But a lot can happen as we transition from in vitro to in vivo experimentation. But in this case, as we transitioned into in vivo experimentation, we saw very similar outcomes. So this is free thoracic vertebra prevalence of Enterococcus cecorum at 42 days. And you can see 17% positive prevalence in the challenge control, which is fairly consistent with a standard SA3 challenge. And then with the 1 pound per ton BIMU, the monoglyceride blend in this case is BIMU, there was a 6% positive prevalence in those samples at 42 days, with the 3 pound per ton statistically intermediate to those two groups. Now, this is the same data, just presented in a different way. And I think it's a little bit more helpful to kind of visualize in this way. So each dot represents a replicate pen. In a complex, it might represent a house or a farm. But in this case, it is a replicate pen. And so just one of these dots in the middle, let's say there's 10 birds, and that might represent 2 out of the 10 birds that are positive for Enterococcus cecorum in the free thoracic vertebra swab at 42 days. And so what we can visualize here is how the dots on the 1 pound per ton inclusion are clustering towards the bottom versus that challenge control over there. And while it's great to see the colonization and data, what we are also interested in is the performance. And so in this case, we did see an improvement in the body weight gain and that 1 pound per ton relative to the challenge control, with again that 3 pound per ton statistically intermediate to those two groups. Numerically, there were differences in feed conversion, but because of the variation, we weren't able to pull out statistical differences in this case. The next products that we've evaluated in combination were a probiotic and a DFM product or products. The probiotic at placement is visualized in this picture here. It was a gel at day 0. And then the DFMs both were in the feed at different concentrations. So you can see the experimental design in this case. We had a challenge control, and then both treatment 2 and 3 received that probiotic gel, as well as different direct fed microbials. And we're going to transition into the colonization data or the prevalence data. starting with the spleens, we have 21-day prevalence, no significant differences, right around a 20% positive prevalence. And then at 42 days, we did see a significant impact with those products on the DFMs that also received the probiotic gel, having significantly less Enterococcus cecorum than the challenge control. Not only was it reflected in the spleen data, we also saw a significant impact on prethoracic vertebra prevalence at 42 days in those same birds. So very interesting data as far as it relates to colonization of those tissues at 42 days. Now, the last thing I wanted to talk about, and I hesitated to talk about it, I have a question mark there just to, provide caution to those as you interpret this. But I have a personal interest because of my background in nutrition and how it impacts disease. And so whenever I have time and space, I try to conduct these small models to look at different interactions. And so as I thought about Enterococcus, I thought about what bacterial challenges might be similar. And I know that this is not going to be a direct comparison, but at least Clostridium and Enterococcus are both gram-positive, right? And so as I thought about products that were effective, nutritional products that were effective against Clostridium, I thought, well, several of the products we've evaluated with efficacy were mineral-derived products. And so I thought, well, might there be an interaction between minerals and Enterococcus? So what I did is I had 3 groups, 25 broilers, or 3 replicates of 2 groups, 25 male Ross broiler chickens in each, and I took them out to 21 days, and I collected 10 spleens from each replicate at 21 days. And so this is the data you're looking at. You can see the challenge control there at a 10% positive prevalence at 21 days in the challenge control versus a 3% positive prevalence in that copper group. So I wouldn't take this to the bank, but I do think it's interesting data and something that ought to be followed up with, whether it's at our facility or somewhere else. So really just to recap and summarize what we've discussed, Enterococcus can be frustrating. I imagine it could be frustrating in the field, given that it can present in a variety of ways. And the way that it presents could be fairly obscure, right? We might see slight increases in lameness. We might see increased culling, slightly increased mortality. And when we do culture, we might not find Enterococcus. And there's a good chance that we're going to find E. coli in a lot of those samples based on what we see at our in our facility. And so it could be kind of a frustrating disease to diagnose. Based on what we see, clinical spondylitis might just be the tip of the proverbial iceberg. I think we see very, very low levels, but there's a really strong impact on body weight gain and feed conversion outcomes in our challenge. And I don't think I did a very good job of highlighting this, but NCSU and SPRG, we really are thankful to be able to use their model, and we try to do everything exactly as they do it in their facilities. But despite that, we see different outcomes in our study, specifically in terms of mortality and prevalence data. And so what that tells me is that there's likely an environmental or nutritional impact on the disease process. And I think that warrants further investigation and could be an added benefit when we think about mitigating this disease. As far as biosecurity is concerned, based on our experiences, it spreads very easily and quickly. And so I think, again, that would have, it might be important when it comes to taking samples of a given house, and also intervention strategies might want to be applied more towards the entire house relative to that challenge. And then as far as interventions, we have evaluated a handful of products, monoglycerides, DFM and probiotics in combination have been effective, and maybe minerals can help. There might be a place for that. But like other diseases, whether it's necrotic enteritis or coccidia or salmonella, I think Enterococcus is going to need a combined approach. I think we're going to have to think about management strategies, nutritional strategies, as well as intervention combinations. We might want to use a monoglyceride early and then transition into a probiotic or a DFM and change the concentration of mineral over time. I think that combined strategy is probably going to be the best way moving forward. As far as what combination, that remains a question to be answered. And so, speaking of questions, I think there's a lot of questions that we could learn more about. Mitsu has described, has really come a long way in understanding the pathogenicity and what genes are important in that pathogenicity, but there's still some questions like, right, why do certain isolates prefer specific tissues, and why are some more pathogenic than others? I think there are still questions that could be answered on that side of the equation. And then the other set of questions that I think are worth pursuing would be, what kind of strategies do we need to think about as far as detecting the challenges in the field, and then how do we apply them and make them useful for our given complex. And then lastly, as far as considerations, as far as when we move forward, I haven't seen the data on environmental and or nutritional impacts, but I think that could be very important as well, in addition to exploring other additives and combinations thereof. So that's all I had today. Thank you so much for your time and your attention, and hopefully either Mitsu and I could answer your questions. Thanks.
Thank you so much. This was really interesting, and there were really good presentations. We have some Q&As from the audience. They're not directly directed to specifically you or Mitsu, so we're open to both of you answering. And the first question that we have here, it's, what point of production should industry focus on reducing Enterococcus load in fields, breeders, hatchery, broiler houses, etc.?
I'm going to stop the share there, if that's okay. Did you want me to keep that up, or is that okay?
It's okay. It's okay.
Okay.
Thank you.
I mean, I should let Matthew answer this as the veterinarian, but I would say the earlier you can stop it, the better off you are, because our experience is exactly the same as Matthew's. It spreads very quickly. We know we can infect one bird per pen and almost the entire pen will be infected. We also know that you cannot have negative control pens anywhere in the same airspace. We end up in our negative control pens, if there are other infected pens, with a 20 to 50, 60% positive infection rate. So sooner, once it's in their house, it's in your house. And I have seen it spread from one house to the next house. And then the byproduct birds are added, and we end up with 2 different strains in different houses. So the earlier, the better. I would say the hatchery and above.
Yeah. I would agree with that. I mean, that would definitely be a place to focus most of the attention. But going back a little bit and kind of back to that summary, I think trying to do a more holistic approach where you're impacting, definitely focus on the breeders in the hatchery, but also I would extend to broilers as well in that scenario until it's under control.
And I totally agree with that, because even a small decrease in the percentage of cases is huge when you think about the numbers of birds. And so not only for the economic issues, but for the animal welfare itself. So I think anything that lessens the disease prevalence is important.
Yeah.
Thank you so much. We have another one here. Why is EC morbidity and mortality lower in British?
I mean,
Great question.
Matthew, you okay if do you want to do it, or do you want?
You go ahead.
These are guesses. They're only guesses. This is theoretical. I don't really know. I think partly I think those birds get colonized much later in the growth period. We know that you can, and once that normal flora is set up, I think it's a few pathogenic bacteria have a lower possibility of escaping the gut. So I think that's one. I think they can become carriers. We often have birds that they have positive spleens, but you would never, ever know that they were sick. And I think it's very easy for a bird to be a carrier, and you just will never know. I would say a good portion of our birds look, fine, and they are positive. I think they're also not on bedding. The wetter the bedding, I think the worse the spread is. So those are just some of my thoughts, and you know more about the production than I do, so I turn it over to you.
Yeah, no, those are all great points. I think that all of that plays into it. So as far as maturation of the immune system and the microbiome, I think are both really important, because we see that in other challenges as well. Like particularly, I'm thinking about cocci right now. I know it's not as relatable, but if we challenge the birds at 21 days, we'll have very different outcomes than if we challenge the birds at 35 or 42. And so I think it has to do with maturation of the immune system and the microbiome. And then also a great point on the breeder management versus broiler management, or comparing those ages. Also we want to think about the density and the bedding. I think that probably makes a really large impact on the disease as well, and potentially even nutrition. But
yeah, I wonder about their more limited diet. That I have no idea what effect that might have.
Yeah.
Perfect. Thank you. We have one more question here. Would you use same methods for control of E. faecalis as you recommended for E. secorum?
I have never done a faecalis challenge. And so let me answer that as I don't know to start with. But I would expect that they would help. Yes, I suspect at the roots that the strategies that we have evaluated would help for both Enterococcus species.
Okay, thank you so much. We have another one here. Does EC persist in poultry houses?
I mean, yeah, you go ahead, then I will.
Okay. So yes. So Enterococcus, and Mitsu mentioned it already, but there is going to be Enterococcus, just like there's going to be Clostridium it's just a commensal. But even talking about the pathogenic strains, for both of those diseases, yes, I think that they can live in the bedding and re-expose another flock.
I know that you've done studies reusing bedding and proven that you can have transmission.
Yes.
And once the houses dry out, like once the birds have been out of there for a long time, the bedding is dry. I can't find it. And I've ground up a lot of darkling beetles and a lot of other things looking for it. And interestingly, when we were doing those longitudinal studies, like we would have a farm that would break and we would, culture it. And then that farm would clear out, they'd set birds again, and we would get it again, but it was not the same strain. So there was another source. Now I assume from the hatchery, we didn't know at the time. So I think it's a combination of all of the above is what happens, but it can, as long as that litter is still damp and you've seen it, you've done that experiment.
Mhm.
We have time for another couple. We have one here. Possible reasons for increase in pericarditis cases. Pericarditis, sorry.
Shall I go for it or you want to go?
You start.
Okay, I only have theory, no fact. I think the different background of those Enterococcus secorums that are causing the pericarditis, those bugs are more visible to the bird. To get that kind of pericarditis, you have to have inflammation. And to have inflammation, the immune system has to be seeing those bacteria. And so I think perhaps the difference in the genetics of these strains, they're overall more visible to the bird's immune system. And I think that's why we're seeing an increase in this early presentation. And I don't know why some birds escape it. Like we all have negative birds, even though everybody's challenged on day one. But that's my theory as to why we see more pericarditis now. The bacteria are just more visible to the birds.
Very interesting.
Very
Yeah, I like that theory.
All theory.
I don't have really a better theory other than just to circle back to we talk about pericarditis strains and pericarditis challenges, but what Mitsu saw and what we've seen as well in that transmission study that I showed you is SA3 tends to create more pericarditis than the pericarditis strains, and so yeah, I'm not really sure what that means, but it might be of interest to those that think they have a pericardial problem and a pericardial strain. It might actually just be a more pathogenic Enterococcus.
Yeah.
Thank you. We have one more here. Can we isolate pathogenic EC or EF from the litter or any other source of monitoring for monitoring?
Well, you could find it, but you're going to have to send it to Mitsu or someone like that that can run the genetic testings to make sure it is a pathogenic strain. Because yeah, I think I'm just thinking out loud here, but if we find Enterococcus in the litter, right, we have to do the genetic testing to make sure it is a pathogenic strain. And we can be a suspect of it when it's in birds, right, because it's in the spleen or in the pre-thoracic vertebra. We can make some assumptions that it might be pathogenic, but really it needs to be tested. But I would say yes, it could be tested for.
Thank you. We have one more question. Should monoglycerides or DFMS be used in the starter grower feed, or should they be considered in all feed types for reducing prevalence of mortality associated with the pathogenic EC and EF?
Okay. So you would want to do those studies, right? Because there are products that we use early that tend to last throughout the grow period. So what we would want to do is we would want to do the study looking at maybe an inclusion of product or products in the starter, and then the products fed all the way through, and make sure there's an additional benefit to having it through all the phases versus kind of focusing it on that most important, the earlier the better, to reference the earlier comment, but try to focus on that early colonization, I think would be the most important. Yeah, you would want to do the study to make sure that is the case.
Okay, I think we have plenty of questions here, but we don't have enough time. So thank you again, Dr. Kirkland and Mitsu. This was an excellent both excellent presentations, and for sharing your knowledge with us. It was really interesting to hear. We also like to thank the attendees of the webinar today for taking the time to listen to our presentation. and our colleagues in the background helping us to organize the webinar. Sorry again for the inconvenience with technology. Fortunately, we made it through and we have everyone back. And before leaving the webinar, I really like to remind you to please take a minute and participate on the survey. It will take you 2 minutes, and it's really important for us to constantly improve on our webinars. Feel free to reach us if you have any feedback or questions, and we'll be sending you shortly the takeaways of this great session. Wishing you all a nice day. Thank you very much, and goodbye for now.
Thank you.
Thank you. See you all.
Bye. Thank you.